Hello, and welcome to Method Development Considerations for the LC-MS-MS Analysis of Drugs. I am Paul Connolly, Senior Product Manager for ResTech's LC Columns. Developing a drug panel LC-MS-MS method for the analysis of drug compounds can be challenging. Knowing how to get started with all of the choices and options can be a daunting task. Starting with a list of analytes, a matrix, and the required detection limits may be all you have. Literature or published methods can be helpful, and vendors in this analytical space can also provide guidance with proof of concept methodology. But it's up to you to make the necessary decisions and selections and ultimately execution of the methodology. Today we'll cover getting started, analytes and goals. We'll look at system requirements. We'll look at silicas for drug testing, phases for drug testing, and even column dimensions for drug testing. We'll go over some modeling and adjustments to the modeling and show you how to add compounds to a method. To develop an analytical method, it's good to understand the objectives that you're trying to achieve. Items such as the precision and accuracy of your method or the resolution between compounds, how fast you need your method to go, what is the desired cycle time to achieve your throughput demands, the sensitivity of your application, how low do you need to see compounds, and of course the reproducibility. Will you be doing recovery samples and calibration curves along with your sets where you need highly reproducible results in order to achieve good linearity and recovery? These are your objectives related to your application. And those objectives require you to make decisions. Decisions like the choice of detector that you're going to use or the sample preparation technique that you will use. We won't be focusing much on those today, but we will be looking more towards the analytical chromatography that you're going to be needing in order to achieve your objectives. And those things come with other choices, the choice of column, the choice of mobile phase, the system conditions that you're going to use in order to achieve these. And those choices should be driven, again, by those analytical goals. Starting with the analytes, we see that small molecule drugs come in many forms and compound classes, but they have similarities that make them amenable to panel analysis. When we look at the structures of the target analytes, we can see similar ring structures and side groups that make them suitable for combination into a single analysis. Here I've chosen a group of compounds that represent a wide range of different small molecule drug structures and types that we can include in this panel analysis for a good example. Next, it's important to understand the analytical system that you'll be using to develop your method. Understanding the specifications of your instrument is a really good place to start. For instance, do you have an HPLC system? That is a system that has a maximum pressure of about 400 bar, large system volume greater than 500 microliters, and is intended mainly for 3.0 and 4.6 millimeter ID columns. Or perhaps you have an intermediate pressure system. These systems are very similar to HPLCs, but they can run a higher pressure, up to about 600 bar, and have lower system volume, maybe around 250 microliters of overall system volume. And those systems prefer 2.1 to 3.0 millimeter ID columns. Then we have UHPLC systems. These systems can handle much higher pressure, greater than 1,000 bar, and they also have much less system volume than the other system types. They could be 100 microliters or less of overall system volume. and they work very well with 2.1 millimeter ID columns. This information, knowing what your system is capable of, can help you to inform about the dimensions of the columns that you would need. So as you can see, logically, we would see larger column formats for the larger system volume and lower pressure systems, while we would have smaller column dimensions for higher pressure, lower volume systems like the intermediate or UHPLC systems. And when we're talking about analytical systems for LC-MS-MS, we're typically looking at these 600 bar to 1000 bar systems with the lower system volume as being best for LC-MS, because we can run these lower dimension columns with lower flow rates, which improve ionization efficiency in mass spec. So if we're putting less solvent into the mass spec that we have to evaporate and desolvate, then we can get higher efficiencies in our ionization. So going to these smaller dimensions like the 2.1 or 3.0 millimeter ID systems and coupling those to LC-MS, we can achieve these lower flow rates. You can see a 2.1 millimeter ID column, its optimum flow rates is around 0.4 to 0.6 ml, where a 3.0 millimeter column is going to require a higher flow rate, 0.7 to 1 ml, and then the 4.6 millimeter columns, a much higher flow rate in excess of a milliliter to about 1.6 milliliters per minute. One of the first choices we have to make for modern HPLC columns is the choice of silica in our column. We now have two distinct choices to make: the fully porous particles or the superficially porous particles. Fully porous particles, as the name implies, has pores or channels that run through the entire particle, whereas a superficially porous particle has a shell of porous silica around an impermeable solid core. The benefit to that is you can get the efficiencies of a smaller particle, but with the back pressure of a larger particle. In comparing these two particles, we can see the approximate 3 micron diameter fully porous particle versus a 2.7 micron diameter superficially porous particle. We can see how the analytes must diffuse through the entire diameter of the fully porous particle in order to get out the other side. So your analytes are entering and they're going through interactions between the stationary phase and mobile phase all throughout this particle and emerging, versus the superficially porous particle, where the analytes can only diffuse through the porous outer layer. The solid impermeable core does not allow the analytes to go through, or even the mobile phase, so they are just interacting with that shell. And looking at a 3 micron fully porous particle, you can see it has to travel through that entire particle versus just going through the shell. In the 2.7 micron particle, that shell is about a half a micron thick, so the diffusion path through it is fairly equivalent to a much smaller particle, even though the size is very similar to a 3 micron particle. And what that results in is faster diffusion through the particle, and we can get sharper peaks out of this that come earlier than the fully porous particle under similar conditions, resulting in a higher efficiency of the superficially porous particle while still maintaining low back pressure similar to the fully porous particle. It results in faster analysis times, and it means that you can get these particles to work on both conventional and UHPLC equipment with similar results. They also handle matrix quite well, which is similar again to the fully porous particle. But you can see we can get improvements in speed without any kind of detriment in pressure by going from a fully porous particle to a superficially porous particle. Regarding stationary phases, what do we usually consider first when we're developing a method? The tried and true C18 phase. Alkyl phases like C18 use dispersive interactions, frequently referred to as van der Waals interactions or London dispersion forces, and are what we commonly think of when discussing hydrophobic interactions on mainly alkyl-based ligands like a C8 or a C18. The interactions are not a result of any chemical electronic bond and are comparatively weak. When looking at neutral molecules, there's a shift in electron density causing an instantaneous temporary dipole moment in other adjacent nonpolar molecules. Hydrogen bonding is an electrostatic interaction between two polar groups that occurs when the hydrogen atom is covalently bound to a highly electronegative atom such as nitrogen, oxygen, or fluorine, and interacts with the electrostatic field of other highly electronegative atoms nearby. In acidic conditions, the surface is neutral and the hydroxyl group on the surface of the silica, the silanol, can act as a hydrogen bond donor. An electronegative atom, such as oxygen, attracts the electron cloud from the hydrogen on the silica, leaving a positive partial charge. The bond results when this positive charge attracts a lone pair of electrons on another heteroatom, like nitrogen. Conversely, in neutral pH and basic conditions, the surface of the silica is deprotonated and the oxygen on the surface can act as a hydrogen bond acceptor. This leaves us with the target analytes for a C18 column that include hydrophobic and acidic compounds that retain on hydrophobicity. Another option is a phenyl phase. Phenyl stationary phases interact with compounds containing aromatic groups or unsaturated bonds through pi-pi interactions. Biphenyl is truly unique among phenyl phases, and biphenyl has greater aromatic retention than traditional phenyl phases due to its two aromatic rings. The two rings are not fused and can be treated independently due to the relatively free rotation about the linking bond. The phenyl rings can rotate around this carbon-carbon bond, which acts as an axis. This allows for more options to conform and connect with structural isomers, resulting in enhanced selectivity over other phenyl phases. So we have looked at retention mechanisms for C18 and biphenyl. Now let's answer the question, why biphenyl? C18 is the most common reverse phase column in use. It's driven primarily by dispersive retention and is great for hydrocarbons and hydrophobic compounds. Biphenyl ligands offer additional, enhanced, and orthogonal selectivity for aromatic species, such as drug-like compounds, making LC-MS-MS analysis faster and more rugged. We state that biphenyl is a good choice for compounds that are aromatic and dipolar or have substituted electron withdrawing groups, fused rings. Let's review some of those terms so we can spot a good candidate for compounds for biphenyl retention. As I mentioned, biphenyl is great for aromatic retention, but what does it mean to be aromatic? First, the compound must be cyclic, it must be conjugated, and it must have pi electrons. Starting with dipolar compounds, these are electrically neutral molecules carrying a positive and negative charge. For example, phenol is a dipole, but benzene is not. For unsaturated compounds, these are molecules that contain double or triple carbon-carbon bonds. Examples of phenol and benzene are both unsaturated. Conjugated compounds, these are species that have a pi orbital on an atom adjacent to a double bond, or a compound with alternating single and double bonds. For example, benzene is a conjugated compound with 6 pi electrons. Fused rings are a system where each bond is part of a ring, where each ring is fused to at least one other ring, and where no bond is common to more than 2 rings. Naphthalene is an example. Electron withdrawing groups have an atom with a slight positive or full positive charge directly attached to a benzene ring with a negative species attached to that. Examples of electron withdrawing groups are CF3, COOH, or CN. This is due to the electron withdrawing groups pulling away electrons from the carbon, creating an even stronger positive charge. Benzonitrile is an example of a compound with an electron withdrawing group, as are nitrobenzenes and nitrotoluenes. Now that we've looked at items like our system and its specifications, particles, stationary phases, and even analyte chemistries, let's talk about our analytical goals for this method. We're starting with an instrument for LC-MS-MS with a 600 bar maximum pressure HPLC that has about 250 microliters of system volume or dwell volume. System volume and dwell volume are the same thing. Basically, the volume of liquid that is held in the tubing and fittings of your system between your pump and your column. Having a lower system volume means you can run faster gradients because the change of your mobile phase at your pump will be felt at your column faster with lower volume. We also are looking at a 17 component mixed drug panel in urine as a matrix, and we're looking for adequate resolution with overall baseline resolution of any isobaric compounds. Baseline resolution is described at a value of 1.5 between closely eluting compounds. We want a runtime of 10 minutes or less. And since greener is better, let's try to limit the usage of acetonitrile in this separation. For the purposes of demonstration, I'm going to be utilizing the ProEasy LC chromatogram modeler for the examples of chromatography. The ProEasy LC chromatogram modeler is a free tool that's available for use on restech.com that is a virtual chromatography modeling tool. With it, we can choose compounds, choose columns and dimensions, and model the chromatography of those analytes on those columns. We can alter the conditions and see how those alterations will affect the overall chromatography. And then we can translate that chromatography to an actual analytical system with a high degree of correlation between the modeled and the actual chromatogram. Accessing the ProEasy LC chromatogram modeler is very easy. It is available on restech.com. From the home page, you just scroll down to the ResTech Resource Hub. There you will see our Easy Suites and calculators. Choosing that will take you to our Easy Suite online software page where you can access our Easy GC tools, where we've been modeling GC separations for almost 2 decades, and our new Easy LC tools where we are now capable of modeling chromatography for LC compounds. It's easy to get to. Just choose the Go to ProEasy LC chromatogram modeler from this page. It does require a login with restech.com, but it is free to use. The login provides you with a storage space within ProEasy LC to save your modeled separations for future use. The first step in using the ProEasy LC chromatogram modeler is to choose the library of compounds that you would like to model. The current selections involves drugs of abuse, for which we have 311 compounds and growing. This includes 80 isobaric compounds within that library. We have a library of 60 PFAS compounds, including 3 bile acids. This library allows for the modeling of many of the current regulated methodologies. We have a nitrosamine library of 16 compounds for pharmaceutical analysis. And we have a cannabinoids library of 46 compounds. So a highly usable cannabinoids library covering many of the regulated, monitored, and even novel cannabinoid compounds. The selection of the library will then provide you with the list of compounds that you can choose from to do your modeling. The first step in modeling with ProEasy LC is to go to the Compounds tab, go to your compound class, and pick your library. I'm choosing Drugs of Abuse, and then from the Drugs of Abuse, I'm going to select these 17 representative compounds. To add compounds, you can just go to the search box and start typing in the name, and those compounds that have a similar name will be selected, and you can choose them. When we choose them, the analytes will turn blue on the list, stating that they've been chosen. You can also search by aliases or CAS numbers to find the correct analyte. As analytes can have multiple names, the CAS number is the most accurate way to find the exact compound. So choosing these compounds, they have a variety of different structural components to them, but there are similarities within those components. We can see some similarities here in these opioid type compounds. Lots of fused rings, electron withdrawing groups, unsaturated bonds are being presented there. You can see these similar compounds where the rings aren't fused, but also aromatic character is provided, which is common for many drug compounds. Here's some other similarities on these THC like compounds, aromatic compounds with hydrophobic tails. We can see some other similarities here with these compounds, and again, additional similarities shown here. Starting my separation with a C18 column and acetonitrile as the mobile phase, I selected a 100 by 2.1 millimeter ID column with a particle size of 2.7. This is a superficially porous particle, as shown by Raptor. Raptor C18 is superficially porous. And the mobile phase is water with 0.1% formic acid and acetonitrile with 0.1% formic acid. I'm starting the gradient as weakly as possible, 97% water mobile phase to start with. As an alkyl stationary phase, C18, I cannot go to 100% water, as this phase would de-wet, meaning the pores would be filled with water and inaccessible by the analytes, causing a loss of retention. So about 3% organic is the minimum that you would want to put on a column like this. And what I can see from that separation to start with is that even at this low organic percentage, my first analyte, nicotine, is coming out in my dead volume. So the T sub 0 value, anything below that is unretained. So you can see my T sub 0 value is 0.41, and that is where my nicotine is eluting at 0.41 minutes. As you can see, the compound nicotine, it is very hydrophilic, so it's going to want to really stay in that water and not interact with the stationary phase that much. Additionally, I have a couple of doublet peaks showing up. Hydromorphone and acetaminophen are closely eluting, as are ketamine and norfentanyl. Now, this is mass spec analysis, and the masses for these compounds are different. So the mass spec will be able to clearly tell these apart. This is not a bad thing, but I would prefer a little more retention between closely eluting peaks. Now, in concerns with isobars, there is a pair of isobars, morphine and hydromorphone, that share the 286.2 molecular weight precursor ion. So we need to be able to differentiate those chromatographically in order for the detector to tell them apart. They are well resolved at a resolution value of 2.9, so there's no concern there. Really, the main concern with this separation is that nicotine is unretained. Since I can't go any weaker with the amount of acetonitrile that I'm putting on that C18 column, I can switch from acetonitrile to methanol as a weaker eluting solvent. So methanol has a lower elutrification value than acetonitrile, so it is a overall weaker solvent in reverse phase chromatography. And you can see going at 3% methanol that my retention for nicotine has improved. So now I am outside of the dead volume on the column with nicotine. And my overall separation is much better. I have good separation between all of my components. There's the nicotine retention outside of T sub 0. But I do see norfentanyl and heroin are close together in this separation. Again, they're not isobaric, so it's not really a concern. But I did want to have a better overall resolution between compounds. And you can see just switching the C18 column to methanol from acetonitrile really improved chromatography overall, including that heightened retention for nicotine. Now looking at the biphenyl phase with acetonitrile, I'm using a similar dimension column, 100 by 2.1 millimeter, superficially porous particle Raptor biphenyl in a 2.7 micron particle size. And I'm using water with 0.1 formic acid and acetonitrile with 0.1 formic acid as the mobile phase. Column temperature is 50 C, and we're looking at starting at a 5% organic gradient and ramping that up to 100% in 6.4 minutes. Overall, the separation looks good. My nicotine peak is out of the dead volume, so it is being retained, even in acetonitrile. And I do have, though, some coelutions there. Peaks 3 and 4, acetaminophen and hydromorphone, are partially coeluting, and fentanyl and PCP are totally coeluting in this separation. Again, these aren't isobars, so it's not mission critical that they be separated. I just have a preference for separating these compounds. The isobars in this separation are well separated. Morphine and hydromorphone have a resolution value between them of 1.8, noting 1.5 is baseline separated. So I have good resolution between those compounds. Just a little coelution issue between a couple other pairs of compounds. Now switching the mobile phase on the biphenyl column from acetonitrile to methanol, I get some good improvements in the overall separation. As you can see, my isobars are still well separated, 2.3 resolution value between those compounds. Peak shapes are good. I don't have any coelutions in my compounds. My runtime is within the 10 minutes that I was looking for. And my overall back pressure is well within the specifications of a 600 bar system. So overall, this is a perfect separation for what I was looking for in my goals. Additionally, it is methanol rather than acetonitrile, so that's that greener separation. You may be curious as to why the separation with methanol on the biphenyl phase looks different than the separation with acetonitrile. And we will talk about that in a moment. In looking at the retention of the analytes on the different stationary phases with different mobile phases, we can see that there are some differences. Now looking at the C18 between methanol and acetonitrile, we can see that the elution order of all of the compounds remains the same, whether we're within methanol or within acetonitrile. We do notice the power of the solvent in the retention of the compound. So acetonitrile will have somewhat less retention on the C18, where the methanol will have more retention. And that's just due to the strength of that solvent. But when we look at the comparison on biphenyl, we have some noted differences. We are seeing some changes, some swapping in the retention profile for some of the analytes. The reason this happens is because when you switch from an aprotic solvent like acetonitrile to a protic solvent like methanol, you are actually activating the pi-pi interactions on that biphenyl stationary phase. It's not that the pi-pi interactions turn off, it's just that when you run with acetonitrile, acetonitrile has pi bonds in it, and you are just crowding the space in your separation with pi bonds, meaning that the pi bonds on the biphenyl phase can't find the pi bonds in your analytes because of all of the acetonitrile pi bonds that are present. So switching to a protic solvent like methanol gets those extraneous pi bonds out of the way and allows the biphenyl stationary phase to interact with the pi bonds in your analytes. And when we look at compounds like acetaminophen and morphine and ibuprofen and meloxicam, structurally we can see why when we have that pi-pi interaction happening, that morphine will retain longer than acetaminophen. There are more pi bonds for it to interact with and retain. The same goes with ibuprofen and meloxicam. Meloxicam has more opportunities for pi bonds than ibuprofen, so we're going to get slightly more retention out of that in methanol versus acetonitrile. We also see, of course, differences between biphenyl and C18 in terms of retention order, and biphenyl and acetonitrile approximates the C18 separation a little better than biphenyl and methanol. Again, that's because we are in acetonitrile with all those pi bonds. That biphenyl stationary phase is using more dispersion retention than aromatic retention to retain the compounds. Now that we've established that biphenyl is a better stationary phase for the retention and resolution of these compounds, and that methanol is a better solvent than acetonitrile for the resolution and retention of these compounds. We can now look at some of the other variables, variables such as particle size. What does that do to the retention and resolution of these compounds? The use of the ProEasy LC chromatogram modeler is a great way to evaluate these. I was quickly able to screen the column phases and solvents, and I can also screen the particle size and see what that net effect is going to be on my overall chromatography. Here I switched the column dimension from a 2.7 micron SPP particle to a 5 micron SPP particle, both Raptor biphenyl. You can see the 5 micron particle has overall less efficiency, and less efficiency means less retention. It means greater peak widths and less resolution between isobars compared to the 2.7. But it also means less back pressure. The larger particles are going to generate less than half the overall back pressure of the 2.7 micron particle. And you can see I still have adequate resolution between my isobars, but it's less than on the 2.7. So morphine and hydromorphone have a resolution value of 1.8 on the 5 micron and 2.3 on the 2.7. You can see generally retention times are similar going through, but peak widths are greater on the 5 micron particle. So ProEasy LC will approximate actual peak widths that you will obtain. The peak shapes are fabricated to look like peaks, but the peak widths are a fairly true representation of what you will see on your analytical system. And as you can see, the peak widths are greater by about 0.01 minutes on the 5 micron versus the 2.7 micron particle. Now going the other way to a smaller particle, a sub 2 micron UHPLC size 1.8 micron particle, everything else being the same column dimension, same Raptor biphenyl superficially porous particle, just going down to a smaller particle size. We can see that the 1.8 micron particles provide more efficiency, therefore more retention and narrower peak widths, and more overall resolution between isobars as compared to the 2.7 micron particle. You can see we start with 2.3 resolution value for the 2.7, and that increases to 2.8 on the 1.8 micron particle. Peak widths are narrower just by a little bit, but they are measurably narrower on the 1.8 micron particle. We're also seeing that retentions are slightly longer on this particle, given the smaller particle size and the higher efficiency. But that smaller particle size also translates into more back pressure. So going to the 1.8 micron particle from the 2.7, the pressure is now about 597 bar versus 328 bar for the 2.7. That would tax the high pressure of my system. Given that it's a 600 bar system, that is not a lot of room to work with. So I would avoid this particle for this separation, due mainly to the back pressure generated. Additionally, with smaller particle columns, the frit that holds the silica in the column is going to be a smaller diameter. On our 2.7 micron columns, that frit is a 2 micron frit, whereas on our 1.8 micron columns, that is a 0.5 micron frit. Smaller frits mean the column is more prone to clogging. So you have to be much more considerate with your sample prep to avoid that you have less particulate matter in your sample, because that will clog your frit and ultimately spoil your column. So when you go to these smaller particle columns, you have to make that consideration in sample prep, but also be aware that you might not have the same longevity of a column due to the smaller frit size and the intraparticle size as well. Another variable that we can look at with the modeling software and method development is the effects of column dimension. So I optimized this method on a 100 by 2.1 millimeter Raptor biphenyl with a 2.7 micron superficially porous particle, and methanol as my mobile phase, water methanol with 0.1 formic acid. And if I just go in and change the dimension in the modeler, I can see what the net effect of going to a 50 millimeter by 2.1 millimeter column would be. Overall, it's pretty good. I have adequate resolution between my isobars, a little bit of coelution coming in between a couple of pairs of peaks there, but generally not a bad separation. Definitely workable. I used the Pro Easy LC method translator, which is a separate tool in our Easy suite of chromatography modeling tools, and I calculated the gradient change going from a 100 by 2.1 column to a 50 by 2.1 column. The method translator is a great little tool. If you want to go just from one dimension of a column to another, you simply tell it what dimension of column you have and your gradient, and then tell it what dimension of column you want to go to, and it will calculate the adjustments to your gradient necessary to achieve similar chromatography. I did increase the flow rate to sharpen the peaks up a little bit. Overall, the separation is pretty good. I've got good separation between my isobars, a couple of doublets that are less resolved than I like, but generally overall workable chromatogram. Now trying a different dimension, looking at the internal diameter adjustment of the column, I went from a 100 by 2.1 millimeter column to a 100 by 3 millimeter column, all other things being similar. Again, I used the Pro Easy LC method translator to calculate the gradient change, going from a 100 by 2.1 to a 100 by 3 column. And I increased the flow rate a little bit to sharpen the peaks. The net result is a beautiful chromatogram. It meets lots of my requirements. The overall resolution between morphine and hydromorphone is even improved with this, kind of owing to the consideration that higher linear velocities, higher flow rates on SPP columns produce highly efficient chromatograms. No peaks are overlapping, good resolution all around, good peak shapes, even more narrow than we saw on the 100 by 3 with the 0.5 mil per minute flow rate. But that flow rate is the kicker here, in that that's increased solvent usage. So I went from 0.5 mils a minute to 1 mil a minute. Chromatography is beautiful, but I will basically double the amount of solvent that I will use. So that would increase my overall cost on this analysis. So still a highly effective chromatogram, and the overall back pressure on this is still also very good. Even at a 1 mil per minute flow rate, the back pressure is similar to what I had on the 100 by 2.1 column at a 0.5 mil flow rate at 326 versus 328 bar. So still a very good and workable chromatogram, and an option if that is what you choose to use. Lastly, I'd like to address adding a component to an existing method. This is, again, something that the chromatogram modeler tool really excels at. You have an existing method, and now you need to add a compound to it. What do you need to do? Do you need to recreate the method from scratch, or can you just slot this compound in with the rest and work it with the existing methodology? Easy way to find out, you can, one way is to just model that singular or added compounds under the same conditions as your existing method, see where the compounds elute, assure that there's no interferences there from other peaks that concern you. You would also have to look at the precursor and quantifier ion in order to determine if this compound is an isobar that would need to be chromatically separated from any other peak in your separation. But looking at this, I can see that the retention time comes out, in a nice break between a couple of other compounds in the existing separation, and it is not an isobar with any of the other compounds in my separation. But perhaps the best way to do it is to just model all of the compounds that you're interested in, your original 17, add your naloxone compound to it, and chromatograph the whole thing. That way you will see if there's any isobars in the way and if you have any overlap. As you can see, just adding this compound to my existing separation showed that it would work quite well on my original 100 by 21 millimeter column, Raptor biphenyl, 2.7 micron with a water methanol mobile phase system. So the gradient works, and I can easily add this compound to my method. So to conclude, producing acceptable chromatography for a drug panel can be simplified. Understanding the specifications of your analytical equipment and the structures of your analytes helps immensely to guide in your decisions. The ProEasy LC chromatogram modeler is a valuable tool for developing these LC-MS-MS separations. And the use of superficially porous particle columns can speed up separations without sacrificing resolution. And finally, the ResTech biphenyl phase is the ideal stationary phase for drug panel analysis.